Okay, that's good. No problem. Yeah, yeah. However you contact me with the link, however that's done, that's fine. And like I said yesterday in class, as far as the field notebooks go, what I'll do is usually within a day or two, within a day or two, I'll let you know if your link works. Can we get it? No, sorry, Marie. There's no more. There's really no more opportunities left. I don't usually like to, do the extra credit. You bring me a flower. I usually like to make it something a little more substantive than that. So, but there's no events or anything like that left. You still got, but remember that field notebook is worth a lot of points, and I just graded. Oh, I just graded the other classes field notebooks, and I would say in many cases it changed a person's letter grade. So don't if you did a good job on that field notebook, that's really going to be helpful. So, and I will get to those grading them today, tomorrow. I mean, probably be middle or toward the end of next week before the grades come back. I will do my best to try to get it back before the final. When is that again? Oh, let's remember. Minimaster screws me up when it comes to. Okay, your final, the lab practical is Monday, and then Thursday is your final lecture exam. So I will try my best to have your field notebooks graded by Thursday, next Thursday. I have to have them graded by, I don't know, whenever grades are due. I think they're the following week, actually, but. Okay. Oh, also, in the interest of time, I'm going to do something here. I don't want to rush through these last few chapters. There's a lot of really good information here. And this mini-master, for some reason, kind of took me by surprise, and I feel like I'm a lecture or two behind. So in order not to feel rushed, what I've done is I've changed how I'm going to present the lecture. For example, if you look in the recorded lectures section now on Blackboard, where all these lectures are, I've taken some lectures from my other class, my other section, which because of the way the mini-master works, we actually just lectured on pretty much the same material last week. And I posted those recordings. Okay, so when you get into the recorded lectures, all the way at the bottom, you'll see something that says chapters 33 and 34. And then in little text below, instead of the date where you usually see, it'll say listen to first. And then I got another one that says chapter 34, and that says listen to last. So I want you to listen to chapter the 33 through 34 first. And then what that's going to do is that's going to pick up where I'm going to end off here today. See, I'm only going to do a part of the insects today. I'm going to stop when you get to the part that talks about insect mouth parts. So you'll see that in just a second. Now, that's on the recording. So you go to the recording for that, and you listen to that recording. And then tomorrow, I'm going to talk to you about mammals. All right? And then the second recording, you'll listen to last, and that'll have all the information about the animals. Okay? Today's lecture will be the one that will fit in between. I'll make it as obvious as I can if this is not making sense, and I'll talk about it again in class tomorrow. But I'll try to make the annotations in the recorded lectures like listen to this one, then listen to this one, because we're kind of going out of order here. But let me not waste any more time on that. If you have questions on that, don't worry. Just take a look on Blackboard, and I think it'll make sense, especially after I post the lecture today. All right, so we were talking about this wonderful group, the arthropods. I mean, if you're not an insect person, then insects kind of creep you out and spiders kind of creep you out, then yeah, I get it, whatever. I understand all that. It's like I always said, I'm not someone that can sit in my house and watch cockroaches crawl around. I don't particularly care for cockroaches in my house. But there's one thing about the arthropods, no matter which type we're talking about, you gotta really appreciate. We saw it yesterday in lab, right? Was all the different things they can do with those appendages. And remember, that's all tied into being segmented, right? With segments, you can have segments with different types of appendages. So this really takes that whole thing that happened in the earthworm, right, with the different digestive parts, and really expands it. And you saw yesterday when we dissected into this critter, there was even segmentation, like, up in the front, excuse me, was the brain and the mouth and all that sort of stuff. The tail, Josh, I think you were dissecting open the tail, or somebody was. It's just pure muscle. So that particular set of segments would be involved in, in movement. Some segments would have the walking legs, the mouth parts, and all that things we talked about, right? As I mentioned too, what happens in the case of the actual crayfish yesterday, what you snipped open was the thorax. Yeah, this piece here. That's what you pulled up, right? Remember, the heart was right underneath it. And what'll happen sometimes with these segments is segments will combine. So you get segments upon segments. You get say, I don't know, super segments, and that's what we mean when we say something like the cephalothorax. And just like what we're talking about in this whole idea of segments, what happens in a tagmata, that's called when you fuse those segments together, you get something called a morphologically functional unit. All right, and that's kind of cool to me, because that's kind of like when we're talking about going from cells that are by themselves to cells that are colonies and cells that are tissues and all that sort of stuff. We're kind of seeing that even at this organismal level, right? So that cephalothorax of that crayfish that we dissected, right, had the stomach, all the brain and the sensory organs, all the muscles to move the mouth parts. And then, that was what we mean. That was a functional unit. That was a food gathering, sensory input, sensory analysis type of place. Okay, the cephalothorax. Spiders, and again, the names kind of change, but spiders, many of their segments had fused into things we call a prosoma and opistosoma. I think that opistosoma, put my glasses on, is spelled wrong. It is S-O-M-A, those two words end in, all right? And again, that's just different names for different parts. So what spiders have done is taken a bunch of segments and basically combined them into two major segments, a front and a back. Pro is front and opisto is back. We all know about the exoskeleton because you learned about it in Bio 1. It's made of chitin. It is a sugar, but you can't eat it, right? It's one of those undigestible carbohydrates. It's kind of like cellulose. There's a little protein mixed in there, and of course, just like we talked about in the bivalves, this exoskeleton is really good for protecting them from drying out, which would have been especially important for the groups. And of course, the insects are the big ones that I'm talking about here, the groups that moved out on the land. Because just like in the plants, right, it's all about the drying out. You can't live on land if you haven't figured out some way to deal with the number one problem of living on land, and that's desiccation. Some of these critters undergo something called ecdysis, right? Molting. I showed you that in a picture earlier, didn't I? Maybe I'm going to get to it. Blue crabs, insects molt, many species do. So if you have your exoskeleton or skeleton on the outside, then in order to get bigger, you've got to get rid of that exoskeleton. So you have to shed that molt. I remember as a little kid, oh my God, in Texas, the locust population would just boom every 7 years, and they would hatch, and there'd be locusts everywhere. My mother absolutely hated it. You drive into a carport at night, the lights, and it would look black on the walls. There'd be so many locusts. Well, they begin molting, so there's all these exoskeletons on the trees and everything around where I live. And I remember my cousin and I used to love to just irritate the crap out of about her sister, my other cousin, because she was really afraid of bugs. And we'd take the exoskeleton and chase her. Ah! Like, there's nothing in it. It's not the bug. But if you ever seen one, you know they look like them. Okay, and the word arthropod, that's another big important part about this. They can move these appendages, right? So they can move around, they can extract, they can retract them, they can extend them, and they use little tiny muscles in that picture you see there. Remember the number one thing about muscles, don't ever forget this, muscles can never push, they can only pull. So in order to move an appendage, in order to flex and extend an appendage, you have to have what we call antagonistic muscles. You got to have the muscles on one side. So for example, when it's flexed, the blue muscle that you see up in the top, all right, that muscle is going to be relaxed. And then the red muscle, that's going to be contracted, so that's going to pull the leg in. And then when opposite it happens, it extends. It's kind of like, again, my bicep and tricep, right? My arm closes because of my bicep, and it opens not because my bicep pushed it, but because my tricep pulled. Muscles only pull. All right, and of course the modified appendages that we talked about in the open circulatory system, right? We can take those appendages, like I said, and modify them in many different things, and that is really sort of the hallmark of what's going on in insects, okay? So we'll go back to the appendages in just a second, but man, this is really cool. We got to talk about this. How do they see? I think about this all the time when, oh yeah, because there's a lot of the chrysalis. You guys probably get a lot of butterfly chrysalis over in your area too, right? Wandering over, I would think. Those are neat. Oh yeah, heck yeah, man. That'd be cool. Well, anyway, got a dragonfly in my back porch this weekend. It's not unusual. Dragonflies get in the porch, and I always think about the poor creatures, what they must think or what they must perceive when they come up against the screen, right? Because we're going to take a look in a second. They really can't see the screen. As far as sight goes, the sensory organs that are available in arthropods, among insects, for example, many insects have ocelli. Now, an ocelli is just like that thing in the flatworm. It's just a light versus dark sensing organ. It's not going to give you vision. It's going to tell you shadows. But what's really kind of cool is in some species, the ocelli will form themselves in a triangle called an ocellar triangle. And if you think about why a triangle, well, that really helps them triangulate where the light or dark source is coming from. So again, we got no vision like we think about vision, but we do have light sensing. And now we have a very accurate way to detect where the light's coming from. If you've ever been out on a boat and you're taking a fix in the ocean, it's always radar or through sight. It's always good to triangulate 3 fixes, right? That's a much better fix when you get 3. That's the same thing with the ocellar triangle. Some only have an ocellus, like the grasshopper bug. But in terms of the eyes, they have these things called compound eyes, okay? And compound eyes are called that because they're a bunch of, and I'm going to move down to the lower right of the slide, a bunch of ommatidium. See all those little things on the right? See those green things in the compound eye? Each of those is an ommatidium, which has a lens, a cone, which is really cool. It's like the condenser on your microscope. It focuses the light, passing that down into the rest of the cell where the light is sensed. Now, what it looks like in real life, I've got some pictures up above. I got a bigger picture of the surface of an eye, and then this really cool picture standing back from that insect right there with his two big compound eyes. Now, back to my buddy the dragonfly. Why is he having probably a brain explosion? Well, because those compound eyes are really bad for detail. I found a cool picture online of what Charles Darwin would look like to an organism with compound eyes. So you see, kind of a face or whatever. Not very good for detail, but great for motion. Great for motion. And a lot of times these insects, especially dragonflies, are predatory. So having that ability to sense motion really good and respond to it very quickly would be something that you would expect evolution would probably drive in these organisms. But detail becomes lost when you have these ommatidium that allow you to really see motion. So my buddy the dragonfly there, while he could perhaps sense the outside, he can't see the screen that's separating them. I was thinking this going, oh, what the heck? There's a force field here or something like that. They do hear, many arthropods, insects, like in this grasshopper, have a tympanum. A tympanum is, did we get to spiracles? No, that's next. See little holes here called spiracles? Those little holes, I'm going to show you in the next slide, is how these organisms respire, the land-dwelling critters, right? If you're in the water, you can use gills and things like that. But if we stretch a membrane over one of these holes, these spiracle holes, we get a tympanum, right? It's a drum. So if you think about those spiracles as being pipes that are led into the grasshopper's body, and, as I said, this is where the oxygen and gas exchange occurs, and have lungs like we do. But if you put a membrane over it, you could make this a sound reverberating organ, right? And so they can hear. Now, their hearing is equivalent to kind of way they see. All right, so they can't say, oh, yeah, that's Pink Floyd versus Black Sabbath, right? They can't tell that. They can just hear the noise. They just can't tell what it is, even if an insect could tell those two bands apart. All right, this makes sense? Okay, those spiracles. This is really kind of cool. This is really one of the things that led to insects just going crazy on land, folks. If you're a marine arthropod, you go with the gills. Gills work in the water. They don't work on land, okay? If you're tiny enough, you can use diffusion, all right? Same thing that the moss is used and all that sort of stuff. But if you're a little bigger than diffusion and you're not living in the water, then you're going to have to deal with gaseous oxygen, right? The oxygen in the water is still gas, but it's dissolved in the water. You're going to have to deal with oxygen as a gas in the atmosphere. So you're going to have some way to move it in and out of your body. Now, you can do it with lungs, for example, is a good way to do it. But these guys don't have lungs. What they have are these little pipes called trachea, just like the thing in your neck is a trachea, just like the ferns were tracheids. Remember that? So these trachea extend from the spiracles. There's the spiracles. They even have a valve on them too, by the way, the spiracles, so they can open and close. Control water loss. It's like a stomata in a plant cell, right? So the air diffuses into the trachea, into the tracheoles, into smaller and smaller and smaller tracheoles, et cetera, et cetera, et cetera, et cetera. And before you know it, what you've got is a tracheole that's small enough and near enough to any cell in the grasshopper's body that gases can diffuse. So diffusion works even in us. That's what moves stuff across our bloodstream. But if you have these tracheoles, you make sure that you're never too far away from those cells that can't live through diffusion because they're not on the outside. Remember, we said if an organism respires through diffusion, it's got to be very small. You can't get real big. We couldn't diffuse enough oxygen into our bodies from the outside in. We have to bring it into our lungs. It's the same thing with these guys, right? Spiders have this really cool thing called book lungs. Now, that picture down below is not of a spider. That's actually of a horseshoe crab. But horseshoe crabs have a similar thing, limulus. That word there, that's horseshoe crabs, have book gills. They're aquatic. But book lungs and book gills are the same thing. It's layers of thin, highly vascularized tissue. I think I've said this before. In organisms, highly vascularized, lots of blood vessels, very thin tissue will diffuse gases, right? Oxygen, just normally. So that's what these little tiny thin layers, they call them book lungs or book gills because they look like what pages of the book, right? All right, so that's them breathing. How about them excreting waste? Everybody's got to get rid of waste. You got to get rid of the food or you got to get rid of the material that you can't consume in food, and you got to get rid of nitrogenous wastes, right? We get rid of it by urinating, okay? How do these guys get rid of those nitrogenous wastes? What is their excretory system? Well, in aquatic species, here's where gills are very beneficial again. Gills are good for exchanging oxygen. They're also good for exchanging anything else. Things move across the membrane in gills through diffusion, and diffusion is not selective. It's simple diffusion. So if in a gill, there's more nitrogenous waste in the blood vessel in a gill than there is in the outside, the water going through it, guess where the nitrogenous waste is going to go, right? It's going to go out. So if you live in a water environment, once again, your gills can use to exchange things, not only air, not only gases, but anything, right? So you can use gills. Now, there are some fish that do this too, as a matter of fact. Now, terrestrial insects, again, gills don't work on land, use this really cool thing. I should look this up one day. I never have. I always wonder, who the hell is Malpighian? But those are called Malpighian tubules, all right? Now, if you look in the picture of the grasshopper, the part that's in the yellow are the Malpighian tubules, and down below is a little better picture. The Malpighian tubules are those little yellow tubes that are coming off that you see there, all right? And what happens there is salt, water, that's not good, but hold on, I'm not done yet. Salt, water, and nitrogenous waste diffuse into those Malpighian tubules, right? And the rest is the food that's moving down and the feces and all that sort of stuff. Now, back in the rectum, something interesting happens. It's not unique to these organisms, but the salt and the water was reabsorbed. Now, your kidneys do something very same. I always tell folks your kidneys are one of those organs that gets rid of waste products. We know that. But what happens is it seems like if you were to take the garbage out, I know my analogy is a little weird, but stick with me. So every garbage day, you took everything out of the house, everything, furniture, garbage, everything, clothes, all the stuff you wanted to keep, all the stuff you wanted to throw away. You put it out on the curb, and then you took back what you wanted to keep. It's kind of the way the kidneys work. The kidneys filter all this stuff out, nitrogenous waste, you want that to leave. Salt and water. Now, salt and water, especially water, you want to bring back in. So later on in your kidneys, that's reabsorbed. I was talking to Josh before class on animal physiology courses. It's interesting in those courses because you find out the chemistry behind why that works. And sometimes when you do that, you realize what I said about evolution not being perfect. Now, I don't know off the top of my head, but the way kidneys are designed, that is, everything is filtered out, then the stuff we want to keep gets reabsorbed. That might have to do with where evolution was leading. Remember, evolution can't reinvent the past. So maybe why that seems like sort of a going around your elbow to get to your thumb way of doing it, it may have been because of the way things were evolving, the way different parts evolved in organisms to get rid of nitrogenous waste. But of course, if you live on land, like I said, it's all about absorbing that water. So it's a good thing that that happens, obviously. Okay, let's talk about some of the different types of arthropods. I know I always say insects when I mean arthropods because there's so many damn insects in the arthropods. Let's go for the chelicerates first, down at the bottom. Now, what happened there? I'm missing a slide. Oh, there it is. Sorry, lost my place there for a second. Oh, the chelicerates. Chelicerae are fangs or pinchers. That's where they get their names. And so this is the scorpions, the spiders. Horseshoe crabs fit in this group too. They have pinchers, they don't have fangs. Our buddy, the ocean spider, there are marine spiders, isn't that cool? The chigger, the red bug, which you see on the right. If you've ever been out in the woods in Florida, I guarantee you've come back covered in chiggers or what we call red bugs. I live right down the road from Red Bug Lake Road here in Florida, so it's probably a good reason why it's called that. What they've done is they got two basic tagmata. They have a cephalothorax and an abdomen. Now, that gets modified in some of them. Like, for example, look at the spider in the upper right. There's a spider, and then right next to it is a harvestman. Now, harvestman is a fancy name for a daddy long legs. You see online sometimes these things that you wonder whether they're urban legends, and one of them that I heard about was the fact that daddy longlegs venom is actually powerful enough to kill you, much like a black widow, very quickly, except that the fangs, the chelicerae on the daddy longlegs are not big enough to puncture your skin. Yeah, well, that's not true. That's not true at all. What is true about them is some arachnid studying people, some some professionals, some experts in this field don't really consider a daddy longlegs a spider. And the reason why it is one of the characteristics of spiders, they have two sections. They have a cephalothorax and an abdomen. So if you look at that spider on the left, indeed, there's a cephalothorax and an abdomen. How many sections do you see in the harvestman? How many tagmata does the harvestman have? Now, remember, we're talking tagmata, so you might be going, what are those little sections? No, we're talking big sections. So I can see a cephalothorax here and an abdomen, but what do I see here in terms of sections? I see one section, don't I? Yeah, so harvestmen, daddy longlegs, have one section, have one tagmata. Okay. A lot of the chelicerates belong in this arachnid group, and arachnids are not just spiders. It also includes the scorpions and the ticks and the mites and all that stuff. But since everybody loves spiders, and the bigger picture of a spider, the better. Oh wait, let me go back one, by the way. Golden orb weavers, some of you might have in your field notebooks. Banana spiders, or garden spiders, we sometimes call them. You ever get a chance to look at those? If the sun, orb weavers, some spiders are orb weavers. Orb weavers, they spin orbs, webs. That's what that means. Look, cephalothorax, abdomen. Okay. Anyway, they're called golden orb weavers because if the sun hits the web just right, the web has a goldish tint to it. See that? It's beautiful. I've seen a couple of those, like when the sun is coming up out in the woods, coming up through the webs. If you get just the light right, it's beautiful. I can never get a good picture of it. The gold never really shows up. All right, arachnids have 6 pairs of appendages, right? They have a pair of chelicerae, which are the fangs that they use to attach with, and all spiders have poison glands that are delivered the poison is delivered through their chelicerae. All right, that's where the bite comes from. They also have these modified appendages, look like little mini tiny legs right in the front called pedipalps. You see those? This particular species of spider has a really is male, and one of it and its pedipalps are really big. Now pedipalps are sensing. They use them for like touching things, chemosensory, kind of like a nose. So they're sensing their environment. This particular spider in the upper right with the male pedipalp actually uses that for reproduction, passing sperm to the female. So that's why the pedipalps are bigger. 4 pairs of walking legs, all right? So we got a total of 6 pairs of appendages. 4 walking legs, a pair of pedipalps, and a pair of chelicerae. Book lungs, orb weavers have spinnerets, which is an organ right back here that they use to spin that web. And not everybody's an orb weaver. Some are hunters. And of course, this is your typical dog tick. I pulled a few of those off Cooper. We do have deer ticks in Florida, which can cause Lyme disease, and that's a problem, but it's not as bad of a problem. Our deer down here do get ticks, and humans do occasionally get bit by deer ticks. But from what I understand, it's more a problem up in the Midwest. There's more deer ticks spread up around to the Midwest and near animals up there than down here. And of course, we do have black widows here in Florida, and they have that red, sometimes it looks like an hourglass on their abdomen, and then the brown recluse. Now, black widows will kill you by paralyzing. They have a neurotoxin. And brown recluse, what they do is they're kind of like a rattlesnake bite. What it does is it causes what we call tissue necrosis, tissue death. That's that brown spot on that guy's thumb. Tissue death. And eventually, like, you could lose an appendage from this. It just rots your flesh away. Oftentimes, folks who have brown recluse, if they bled it gone a little too far, the way the doctor cures this, he has to get all that dead material out of there. So they'll end up sometimes having a little bit of a crater, like a scar. Brown recluse, find them in Florida. Again, black widows, I see them every once in a while. I used to see a lot of them over at Valencia. But I wouldn't say that... I wouldn't say there's a ton of them. Brown recluse, even less so than the black widows. Now the myriapods, those with lots of feet. Pod, feet, myriad, right? Myriad means a lot. Myriapods have taken this segmentation and just simplified it. Boom, 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 boom. One segment after another. As a matter of fact, that's how they grow. They grow from one, they just keep adding segments as they get bigger. They live on land, so internal fertilization, something you might expect. It's not always the case in organisms that live on land, but internal fertilization is a good way of protecting those very sensitive gametes, right? You don't send them out into this dry environment. You're inside of an organism's body and protected the whole time. They lay eggs, gonorrheids, so there are male and females. There are no aquatic versions of these guys. Centipedes and the millipedes, all right? Here's how you tell them apart. Centipedes have fewer legs, millipedes have more. Okay, it doesn't, centi means 100 and milli means 1,000. Doesn't mean that centipedes have 100. It's just, we're talking in relative, right? So that one on the bottom, that's a Florida centipede. And the one in the upper right, that's an ivory millipede. I'm not sure which centipede that is, but that does occur in Florida. But those ivory millipedes, if you went to, you've seen those all over the place. Those are very common. They're the things that if you're walking along a sandy road out in the woods, you see these little trails that run parallel, like little train tracks all over the place. Those are millipede trails. They're real small. They're probably not much bigger than what I'm drawing there, but those are the little legs moving across. Now, centipedes are meaner. They have poisonous fangs or claws and are carnivorous. Millipedes are herbivorous, but that doesn't mean they can't defend themselves. They defend themselves by smell, right? So there are these paired glands in each segment that if you mess with them, they release this smell. Some even release cyanide gas, which is something that you don't want to be around, obviously. It's very effective, right, for defense. Defense, you can run, you can fight, or you can make, oh, don't mess with me. You can make it offensive for the predator to come after you. I remember a buddy of mine working on vultures, and he told me that what vultures do is if you go mess with a vulture, they throw up on you. Think about what vultures eat, and then they're throwing it up on you. I mean, vomit itself from good, from fresh food isn't particularly appealing. Can you imagine vomit from carrion? So that's probably a pretty effective. I said, you ever been thrown up? He goes, yeah, it's awful. tagging the birds. He goes, yeah, it's being thrown up by vultures is probably one of the worst things you can get thrown up on. But again, good defense mechanism. Oh, that's all right, Josh. It happens, right? Technology is against us. It's the ghosts in the machine. Now, this is kind of new, actually. We used to talk about the insects and the crustaceans and lump them together, but now we're going to call this big group, including a very small group that we don't discuss, the Rumpedians. But we're going to call the insects and then the other crustaceans the pan crustaceans. Pan just means like a big name for a bunch of crustaceans. All right, crustaceans are largely marine. There are some freshwater, like that copepod on the right. That actually is a freshwater crustacean. Look at all those appendages. Those are eggs that are being dragged behind, by the way. That's what those are. Those are eggs. Modified appendages for chewing. They have 3 of those, various pairs of legs, live in the water. They can diffuse across gills, or if they're small enough, across their cuticle. Males and females. Now, here's why we're lumping the pan crustaceans together. Lumping the crustaceans together. All crustaceans have a similar larval form. It's called the Nauplius larva. This is what it looks like. This is what it really looks like under a microscope. It looks kind of like a mite or a tick, right? All crustaceans have that. And remember, I've said to you a number of times, when we think about phylogeny and evolution, if you have a similar larval stage, it usually means you come from a similar or a common ancestor, right? So that's what unites crabs, krill, very important food source, especially down in the southern ocean around Antarctica, copepods, lobster, our buddy the sand fleas. Barnacles belong in this group. Remember, they have a shell, but that's convergent evolution. They're not mollusks. They have actually do have appendages. I'll show you. These little things that we used to call roly-polies, or still do as kids, right? Those aren't insects. Those are not insects. They're crustaceans, right? So there are some terrestrial crustaceans, obviously. In this group of crustaceans, we have those that we call the decapods, because they have 5 pairs of thoracic appendages, or 10 feet. Our crayfish was a decapod. It had 4 walking legs, and then the front leg was modified into a pincher, or a cheliped. Chelicerate is fangs or pinchers, so a cheliped is a pinching foot thing. A ped or a pod is a foot. Exoskeleton, which these organisms essential for survival, good for predator protection, is made typically of that very common ocean substance, calcium carbonate. It's the same thing that makes up the hard part of the coral animal. It's the same thing that makes up the shells of the bivalves. It makes up the tests of the foraminiferans. Remember, this is chalk. This calcium carbonate is like it's like the cement of the ocean world. If you need to build a structure, something hard and rigid, you're going to use calcium carbonate. A little bit of terminology. That thing you pulled off from the thorax, covering both of that, the cephalon and the thorax, is something collectively we call the carapace. It says here that it covers the cephalothorax, but what I mean by that is it covers in terms of organization. That's all the carapace up there. The little legs back behind are the swimmerettes. They're used for reproduction, like we talked about in class, and of course for swimming, and they're just modified appendages. Crabs basically differ from lobsters in one very simple way, if you think about it. A crab is just broader. The abdominal region down here is smaller. It's vestigial. Blue crabs. This is the abdominal region of the female and the male. Blue crabs are sexually dimorphic. You can tell males from females by looking at their abdominal region. Females often in many species carry their eggs on their abdomen. I was telling you about that with the crayfish. That would be where they would carry them on their legs down there. And among the crabs, very often they're going to carry their eggs. So that's why they have a larger abdomen there. All right, so if you take a lobster and you squish it and widen it, you make a crab. You take a crab, you lengthen and narrow it, you basically make a lobster. Oh, barnacles. All right, now barnacles are crustaceans. Barnacles are not... they are not mollusks. They have shells, and that's why we originally thought they were mollusks. But because of the larval form, because of that Nauplius larval, and because of these appendages, they're filter feeders, you see. They have shells, they're sessile. This is the barnacle of a larvae. That's a Nauplius larvae. But what they've done with their appendages, what they modified them into are these things. See how you can see they're jointed. So they're filter feeders. They use these to trap food. And they can stick onto things really well, right? They can stick on the ships. They can foul up buoys and stuff that's in the ocean. Whales. You can see the blowholes of that whale. Barnacles attach themselves to that. We have no reason to believe that harms the whale other than the hydrodynamic drag. Whales tend to have a really thick blubber layer, so we doubt that the barnacles get down. And blubber's not really well, it doesn't have a lot of nerve endings, so we doubt that they get that far down to cause pain, let's say. Of course, like I said, the hydrodynamic drag slows them down. All right, now hermaphrodites, they're male and female, all right? So they could fertilize their own. That's the thing, when you fertilize your own eggs, you lose variability, right? So you want to avoid doing that. You can if you need to, but it'd be better if you could breed with somebody else you're not related to, for reasons that we've talked about a million times. But you're sessile, you can't move. And you want to up the chances of your gametes, let's say you're the male or the sperm, of getting to the eggs, all right, in the other barnacle. You want to increase the chances. Now, one of the ways you can do it, sea urchins do this, is they basically just, the sperm from the male goes out into the water, the eggs from the female go out into the water, and maybe they mix together, maybe they don't. Of course, we've talked about what happens to most of them. Barnacles have the notoriety of having the world's, scale-wise, largest penis. This is a barnacle penis. What they do is, if you're sessile, instead of taking that chance of shedding your gametes into the water, you extend that very long, relatively speaking, penis over to the other barnacle, right? And you release your sperm and fertilize the eggs in that barnacle. Another way of kind of coping with this randomness of what happens to our gametes, right? Especially among a sessile organism. Remember, I said a lot of sessile organisms have a planktonic or a drifting larvae, like this barnacle larvae. But this one approaches the idea, what do you do about the gametes? Well, how can you increase the chance of the gametes get together? So what you do is you basically are engaging in kind of internal fertilization with that world's largest penis. So no matter what you think and how proud you are of yours, if you're male, barnacles are scale-wise much bigger. All right, insects. Hexapods. So they got 6 feet, right? Several orders of insects. Over 30, right? And we won't discuss 30 orders of insects, but this is definitely the largest group. You can see a couple in the background. My buddy the Odonata, our friend the mosquito, fleas and scarab beetles, and all kinds of neat bugs that are out there. Largest group, half of all named species. There's 10 to the 18th insects alive at any one time. I was doing a little math with that the other day. If you want to get an idea of how many that is, calculations from astronomers, so I'm really shifting gears here, estimate that there's about 10 to the 21 stars in the universe. So there's about 10 billion galaxies, each with about 100 billion stars. So there's 10 to the 21. So there's 10 to the 18 insects. So relatively speaking, there's almost as many insects alive on Earth at any one time as there are stars in the universe. It's a lot of critters, man. They've taken those segments and divided up them into, what did I see? Something's missing here. Oh, I see. They got 3 body regions. It's the indentation. Head, so this should be moved over. The thorax, which has 3 segments, 2 legs a segment, sometimes wings. Wings are not appendages, all right? Wings are not appendages. Even though I've said all along that arthropods are all about the appendages, wings are outgrowths of the body wall made of chitin and protein. Doesn't mean they weren't important. During the Carboniferous, when there was all those, remember, it was real swampy, lots of cypress trees and cedar trees, and just like, Florida, right? That was the Carboniferous, if you think about that, all over the world. The evolution of wings in insects during this time really led to the speciation of insects because they're able to radiate out. They're able to move out into different environments. So they got the head, the thorax, and then they have an abdomen. And most of these guys also have compound eyes. So good for detecting motion, not so good for recognizing Charles Darwin. All right, now, I'm going to stop here. I'm going to keep the recording going, but the next slide right here, this one, is in one of those recordings. It's in the part number 1 recording. And so what I need you to do is sometime between now and the time that you have your final exam, so I know you guys got a lot of work to do with lecture exams and lab exams coming up in other classes, so I'll give you a little bit of flexibility here, and also in the interest of time, like I said, listen to the one that says chapter 33 and 34. It's going to start right here. So this is where that part 1, you'll see it, I've labeled it part 1 in the recorded lectures. That's where that's going to start. And that's going to take you all the way through the rest of the insects, through the echinoderms, and into lampreys. So now I'm going to switch slides, and I'm going to pick up where the recording part 1 will stop. And then I'm going to stop where the next recording is going to pick up. I'll label this really well in the recorded lectures. Let me get down to where I am on the fish. So, yeah, right here. There we go. So when you turn on, when you finish the recording of part 1, this is where you'll stop. All right, this is where you stop. This will be the last slide. You'll talk about lampreys. Okay. So what we're going to do now is we're going to move on from here, and I'm going to discuss the fishes. And at the end of the fishes is going to be the amphibians, and the amphibians are on the next recording that you're going to listen to. And the only thing the next recording doesn't have is the mammals, which we'll do tomorrow. All right. Again, I'm going to lay this all out as much as I possibly can in the on Blackboard, so it's not confusing. But do email me if you have any questions. All right. So here we are. We've moved outside of those fish. We're now going to talk about the fish that have jaws. All right. So what we were talking about in the lampreys and the hagfish are what we call fish, but these are fish that didn't have jaws. They had a circular mouth, and they might have had teeth, but they were called cyclostomes. I don't see that word written up here. I see gnathostomes, but cyclostomes, cyclo means round, and stome is mouth. All right, so these are the cyclostomes, are the Myxinae and the Petromyzontida, these two guys right up here. And now we're going to the gnathostomes, and gnatho means bones, jaws, right? It means now you have a hard structured jaw. So we're going to move now first into those organisms. This is the dissection we'll do tomorrow. That first set of gnathostomes we're going to look at are those that have a cartilaginous skeleton. All right, so they have jaws and they have a mineralized skeleton. Now, that endoskeleton organisms can be either mineralized with bony material and make bones, or it can be mineralized with cartilaginous material and become cartilaginous. There are other fish that have a cartilaginous skeleton beyond the sharks and the skates and the rays, the chondrichthyes, but they're the one most well known. As a matter of fact, the word literally means cartilaginous fish. Ichthyes is fish. Chondra is cartilage. All right, so let's talk about these cartilaginous fish. First thing you might think when you think of cartilage is, oh, their skeletons must be weaker, right? Because, I mean, cartilage in your body, it's flexible. Your nose is cartilage and parts of your ears are cartilage. So you think of this soft structure. Well, tomorrow when we get in the lab, you're going to find out that the skeleton of that shark is anything but soft. It's just as difficult to get through as it is a bony skeleton. So don't get confused by the fact that just because it's made of cartilage, that sharks' skeletons are sometimes not as strong as other fish skeletons or other bony organisms are. All right, so the first fishes that we talked about, as I said, were agnatha. They didn't have jaws, and these were the ostracoderms with cartilaginous skeletons. And the only ones around today are the two groups that you'll hear about in the previous lecture or in the previous recording are the hagfish and the lampreys. Now, when jaw evolution occurred into the gnathostomes, that really changed everything. Like I mentioned before, that starts an arms race, right? Because when you begin having an ability, a major ability to overcome your prey, then you're really going to ramp up the environmental pressure on the prey to be able to find some way to avoid or get away from the predator. Well, where did these jaws come from? Where did these hard structures come from? It's kind of an interesting story. Down in the lower right, there's a picture. Gills are, there's the gill slits. Gills are held up by skeletal rods. It gives them some support. What we think back in evolution occurred is a couple of those most anterior skeletal rods, so the one in the green and the red here. Yeah, I like snook. Does taste good. Very, very obvious fish too, that dark line. So what they did is they took these skeletal rods through evolution, and those skeletal rods were modified and changed and positioned and evolved eventually into the jaws of the organisms we have today. And that, again, is what started off that arms race, right? So you have the advent of the jaws. You also have this lateral line that we just talked about. That's what this dark line is down the line of a snook. Many species of fish, I don't know if I want to say all. Is it all? Yeah, hate to always say all, but many species of fish have a lateral line. It's not really a line. If you looked at it more closely, it's actually a bunch of pores. See in the middle there? I've got a cartoon of it. It's a bunch of surface pores with a canal connected to this tube where there are these sensory cells. Lateral line systems sense pressure waves. So that's what they always say. Sharks are attracted to fish that are flipping around doing what we call the dance of death. So this fish is got something wrong with it, and it's putting all these pressure waves, and the shark's like, what's going on over here? And it swims over to it. Or you're kicking in the water, treading water. Shark says, wow, what's going over there with those pressure waves, right? So they can use it to sense prey. Blind cavefish that don't have eyes, we used to have some in the aquarium there at school. I was always amazed at how they could swim around and always avoid the rocks and the edge of the aquarium and all that stuff without ever bumping into it. If you think about it, the way they did it is they swam closer to a structure and they pushed that wave of water ahead of them. It's kind of like radar, right? As they got closer, that wave would bounce back faster at them. And they would sense that with their lateral line, and they would know they're getting close to something. So these first fish, the placoderms, these are the ostracoderms, and these are the placoderms. This is just hello pencil. It's not working, of course. Stracoderms, placoderms. Placoderms are called placoderms because they had these armored plates. And we see these are ancient, these are extinct, these are fossilized. But when we saw placoderms in the fossilized record, we went, there must be something going on here. There must be some big time of predator prey thing going on, because this guy's covered himself with bony plates, a head shield, a thoracic shield, things to protect itself from being eaten, right? And so we look in the fossil record and we see at the same time, even this guy had them, he had strong jaws, right? So the jaws and the protection, they go hand in hand, right? It's that arms race. Now, as we move on to sharks and more modern bony fish, all we've done to those jaws is continue to modify them, continue to improve them, because in many cases, these animals live or die by their jaws, right? That's why sharks are always able to replace their teeth. You lose your teeth, you're not much of a predator. Okay, gills, and we'll be able to trace this in the dogfish tomorrow too, but first of all, at the heart level, fish only have a two-chambered heart. They have an atrium and a ventricle. And the ventricle is the pumper, and the atrium receives the blood. So the way it basically works is the blood leaves the ventricle, it's blue, it's deoxygenated, it goes to the gills, and it becomes oxygenated. Once it becomes oxygenated, it goes back through the body, and it delivers that oxygenated blood to all the tissues and becomes deoxygenated back to the atrium and pumped again. Single loop circulatory system, we call it. As opposed to you and I, right? We have a double loop circulatory system. Our heart has 4 valves. One loop goes through the lungs and the other loop goes through the body. All right, now let's talk about what happens in the gills. I'm not real concerned with you guys knowing all the intricacies of gill structure, but there is something about the function of gills I want you to, if not be amazed by, at least be aware of. So if we looked at the gills in a fish, each of the gills have a filament. Each of the filament have a bunch of lamellae, and basically what we're talking about is really thin tissue that's vascularized. Now, the cool part, the physiology. Look what's happening down here. You notice that the blue blood, which is deoxygenated, flowing to the red blood, which is oxygenated, and you notice the water is flowing the other way. This is what we call a countercurrent system, and we see this a lot in physiology. What this entails is that there's always more oxygen in the water than there is in the blood. And I'm not going to get into the details of this, but as the water flows across the gills, because of the way the blood's flowing in the opposite direction, we're always making sure that diffusion favors oxygen flowing in and carbon dioxide flowing out. It's an absolutely incredible system. It's also very similar to the way fish maintain buoyancy, to a technique that they use. But if you did take a physiology class, you'll learn about this counter flow or countercurrent or counterflow, either way. And to me, it's one of those things, one of those simple things of physics that Mother Nature understood, living organisms did, and just used a bunch of different places. Fish, like the rest of the vertebrates, like the rest of us, have nutritional deficiencies. For example, we have an inability to synthesize certain amino acids. We can eat them, okay, but these essential amino acids, but we don't have the ability to create them, okay. And sometimes what happens amongst our amino acids when we're building proteins is we turn one amino acid into another. So what that means is there are certain amino acids that are in shortest supply in our diet, and one of those is lysine. Lysine is one of those amino acids that, while you can get it from your food, you don't get enough of it, but you need more of it. So your body converts more plentiful amino acids into lysine. If you remember and you wonder why the hell there's a picture from Jurassic Park down there, Marie, Jurassic. Do you remember what they meant by the lysine contingency in Jurassic Park? That was, I took that right out of the book, actually. The lysine it was a way to protect the world from these dinosaurs getting off that island and living in other parts of the world, right? What they had done is they had removed the ability genetically for these dinosaurs, for these reptiles on the island, to synthesize lysine. So where we would have the ability to change one amino acid into another, because we would need it, into lysine, these, as other vertebrates don't, these guys in the movie, in the book, genetically modified these dinosaurs so that they couldn't do that. So how did they keep them alive? Well, they fed them lysine supplements. So the idea, if they escaped to someplace else, up another part of the world, they would not be able to assimilate, take in enough lysine. And since they didn't have the gene to make it, they would have a lysine deficiency and they wouldn't be able to make all their proteins. That's how they got off the island too, by the way. They got off the island and found some crop, I think it was in Costa Rica, that was high in lysine, and went to eating that or something like that. Okay. So enough about the evolution of Jaws, but my goodness, we can't talk about the sharks unless we talk about Jaws, right? That's one of the things they're well known for. They're not all carnivorous meat eaters. They're not all a mouthful of teeth. Look at that. Now that is a gnathostome, brother. Let me tell you, that is a very efficient predator. Lots of really sharp, in some cases serrated teeth that are replaced in a conveyor belt. So if they're lost, you get new ones. You're constantly replacing them because guess what? You live and die by your teeth. Not all of them are predators like that, though. The one in the upper right, the whale shark, that's a filter feeder. Love to die with the whale shark. You get up to 60 feet long, so that's a filter feeder. These guys were very prominent during the Carboniferous. They're still pretty prominent now, but they were much more prominent back about 280, 350, or 60 million years ago. So the cartilaginous skeleton, it's not versus bone, but instead very light, very strong. The teeth, where did they come from? You should ask. If you've ever felt the skin of a shark, did you? Oh my God. No, you did. Oh, people on the internet, really? No, I didn't get a chance to see it. Yeah, I didn't see your other text. I thought you did. I'm like, oh my God. Yeah, I'd love to be able to do that. But if you ever rubbed a shark, they're smooth in one direction and rough in the other. This is a microscopic picture of the scales of a shark. Sharks have special scales we call placoid scales. And they look a lot like teeth, don't they? And that's because that's where the teeth evolved from. We think that the teeth in the gnathostomes came from the modifications of their scales. Okay, so the sharks, the skates, and the rays, this is your Florida stingray. This is a white-spotted bamboo shark that was born at SeaWorld, actually born as an albino, which is rare. We don't often find, considering these are predators that lay down on the bottom and try to blend in, what we call ambush predators. Wait for something a little bit too quick and attack it. This guy wouldn't be able to ambush much, though, right? He stands out like a sore thumb. Oh, and these really cool things called chimeras. Ratfish, they're also called. That's a horrible one. I like chimera better. Deep water. Very prominent lateral line, very big eyes. A little bit of biology detective work here. Big eyes means deep water, low light levels. So that's why we know that these organisms are usually found in deep water. Some species, really interesting enough, actually, some males have a penis on their head. So they can actually swim up behind the female and insert it in her cloaca, which I'll show you what the cloaca is tomorrow. It's an opening. The cloaca has a special terminology for it, though, or a special application. How do you tell a stingray from a skate? Well, for one thing, stingrays give birth to live young, like lots of sharks do, but not all do. Skates lay eggs. There are some sharks that lay eggs, but morphologically, stingrays have a barb right near the base of the tail. And the way it works is it's good protection from above. So if you looked at a stingray from the side, there's the tail. Try to draw like, there's the barb. You step on it, all right, it raises up its tail and jabs that barb into your skin. Now, I'll show you a barb in lab tomorrow, but that's not something you want to get jabbed into. It's got all these recurved hooks on it. Goes in real smooth, comes out really ragged, and usually leaves behind a lot of tissue, which causes infections. And it's got I'll show it to you, it's a lina with a bunch of venom cells. I've never been hit by one, but a buddy of mine did. And he said for 2 hours it had to have been the most excruciating pain he'd ever had. Skates also many times have finlets on their tails, and they have these knobby protuberances that run down the midline too. They look a lot alike, though. The manta ray down below that you see, beautiful. You can find those around Florida. I did a necropsy on one on Canaveral National Seashore many years ago of when I was about 13 feet across. These are filter feeders. They use those big things in the front. They call those cephalic folds to filter the water in. One of my favorite is the spotted eagle ray down in the lower left. Those are in Florida too. I was diving one time, had 3 of them, just kept circling right around me. I don't know if they were interested in me or what, but they were really neat. I love their scientific name too. Etabatus neri neri. That's really cool. Whoops, hit the mute button. All right, reproduction, internal fertilization. Males have claspers, 2 of them, and they'll insert that clasper into the female, and that's how they'll deposit the sperm. That is kind of like the barnacle, right? It does a better job of ensuring that the sperm gets to the proper place. Most members of this group are viviparous. All right, let's introduce these terms. Viviparous, oviparous, ovoviviparous. Viviparous, live young. Humans, mammals are viviparous, most of us. Oviparous, lay eggs. Birds, skates, horn sharks. These are some of the eggs. These are called mermaid's purses sometimes. This one up above is of that white-spotted bamboo shark. I think this is a horn shark, if I remember right. I'm not really sure. But those are the egg cases. I'm trying to actually, that's my hand. I'm trying to show up so you can see the embryo inside. But those are the egg layers. And then the ovoviviparous, or what we call aplacental viviparity. What we mean by that is that the young stay inside the mother until they're older and better able to survive, and then they're born. But she doesn't provide a placental interface. See, if you're viviparous like human beings, there's a placenta. That's why it's aplacental viviparity. You give birth to live young, but you actually lay an egg basically inside of the female's uterus, and then that egg hatches, lives off the yolk that the egg provides, which is what you find in other egg layers, and then the individual is born. Sand tiger sharks are known for being ovoviviparous. They're also known for something called adelphophagy. Adelphophagy, I missed a syllable there, brother eating, or another term for it up above, intrauterine cannibalization, or a cannibalism. Basically what that says is inside the womb, the first one who hatches, right, so she'll lay 3 or 4 or 5 eggs into the uterus. The first one that hatches will actually eat all of its brothers and sisters. So they'll engage in cannibalization. They'll engage in or cannibalism while they're still in the uterus. It's a rough world out there, let me tell you. We worry about sharks conservationally speaking. Maybe a lot of people don't, because they think, well, who cares if all the sharks were gone? Well, if who cares if all the sharks were gone, we'd be flooded with all kinds of other types of species that would unbalance the ecosystem. The problem with the populations, you can see so many of them have been decimated. White sharks down 79%, gray sharks up to 83%. Mako sharks are very open water, so they don't often come into contact with humans, lucky for them. The problem with conservation on sharks, though, is the fact that they have relatively long gestation periods and they produce relatively few offspring. So, they take a lot more care of their offspring, but since they make fewer, that means there's fewer in the environment, and there's greater chance that population declines can become problematic in these guys. Okay, let's go on to the what we call the osteichthyes. All right, so these are the bony fish, and the first bony fish we're going to talk about are the ones that have basically triangular fins. That's what Actinopterygii means. What a cool word, right? Actinopterygii. Actinopterygii, the actin is the rays, the pterygii means a triangular shape. So these are all your other fish, right? And again, evolution worked in many different places at the same time. It wasn't like these evolved after the sharks. They evolved during it. But they have these parallel bony rays that you show in the picture there. Right? I'm showing you in the picture. You pull up the fin of a bass. You know what I'm looking at there. And that supports the rays, but there's no muscles within the fins. And I put that on italics because that is going to become important a little bit later on when we talk about some of the other type of fish that are out there that actually do sort of have these muscles in there. Most fish lay eggs. Some are internal fertilizers. This is mosquito fish, guppy, we call them. And these are internal fertilizers. That's actually a pregnant female, a black dot right there. I used to work with these fish in South Carolina. It tells you that it's pregnant. They have a swim bladder up above. They have a swim bladder, what's used for buoyancy. It's like a buoyancy compensation vest or compensator vest that divers use. So they inflate with air and it rises, deflate, and they sink. And they use that counter current system, very similar to what we saw in the gills, to either release gas through the gas gland, which would cause them to rise, or remove the gas through what we call an oval body, which absorbs the gas. Internal, so they can actually use that to go up and down. Sharks, as we'll find out, don't have a swim bladder. What they use for buoyancy is a big oily liver, which will be a very prominent thing when we cut open tomorrow. Big oily liver oil floats, okay? Another thing that the Actinopterygii have that the sharks did not is they have something that covers their gills. That is called an operculum. Take a look back at the shark. Notice, gill slits, they're open, right? No operculum. Typically, sharks have 5 gill slits. There are some other species, but none of them have an operculum. The operculum is going to protect the gills, and they can use it for bellowing. They can use it for pumping water across their gills, especially when they're sitting stationary. Sharks are ram ventilators. That means the water goes in the mouth and out through the gills, so they very often have to move through the water, although there are some exceptions to that. 27,000 different species of bony fish. So this is a very, very prominent group. Eels are also bony fish. This is the eel I worked on in college. Matter of fact, that's me and my major professor holding one of the largest ones we found. This is about a 6-footer. We'd find these in the Gulf of Mexico. These were called the giant snake eels. All right. We've got a couple of other type of fish, the lobe-fin fish, and I need to discuss those because those are going to take us very nicely into the organisms with legs, right? Because fish don't have legs, but some fish have fins a little bit different than the other types. So these are what we call the actinistian, which are the lobe-fin fish, and the dipnoi, which are lobe-fin, but we also call the dipnoi the lungfish. Down below in the picture there, that's an actinistian fish. That's the coelacanth. Now, there's an interesting story about coelacanths. Coelacanths are alive today, but they're living fossils. We used to think they were extinct. We thought they went extinct with, I think it was the dinosaurs when we thought they went extinct. But we, back in was it the 50s? Maybe the 30s, Somebody down on one of the docks in South Africa where fishermen were coming in happened to know a bit about ichthyology and noticed in the pile of catch there was a coelacanth in there. So they're not extinct, okay? So that's one of the interesting things about them. But the other thing, if you look up in the upper right, those ray fin fish that we just talked about, remember there's no musculature into the fins. It's just rays. There's a shoulder girdle, and then the rays extend out from that. But look at the coelacanth's fin. It's got the rays, okay, but notice it's got these a shoulder girdle with this central core of bones that extend out into the lobe or into the fin. So this is the ray part, and then they've got this bony structure. You know, maybe the beginning of legs. Maybe this is what we talked about in evolution when we talked about what good is 5% of a leg. All right, in this particular case, that's part of a leg. What advantage? Well, this organism can still swim around. So you notice the jump is not all that crazy to go from the humerus of a coelacanth. We have a humerus too, right? So while these organisms stayed in the water, we do see, you know, this beginning to move out on the land, all right? And these guys evolved back when all the other fish evolved, and they've been around a long time. They just stayed in the water, but somewhere along the line, now it wasn't the coelacanth, but it was a very close relative of the coelacanth, we believe is an extinct relative, we think is what gave rise to the amphibian ancestors. There's only one or two coelacanth species around. Now, lungfish have lobe fins too. I've pointed them out with the red arrows, but lungfish also have an accessory organ, kind of like a lung, not a lung like we have, but a way that they can actually exchange air. Lungfish are also an interesting story about plate tectonics, you know, the continents drifting. If we take a look back there in the Permian, the continents were all together like you see on the left, and lungfish were found all throughout this region where it was very tropical at that time. Antarctica was very tropical. As the continents moved away, the lungfish went with them. So today we find lungfish in South America, Africa, Australia, and fossilized extinct ones on the continent of Antarctica. Looking at such a specialized fish like that, keeping in mind the law of the parsimony, we went, what are the odds that lungfish evolved in these 4 different places independently? That's less parsimonious. It's more parsimonious to assume that they all lived in that one central area and then they just kind of drifted with their continents. So that's what we call biological evidence for continental drift, right? Okay, just a couple of more slides. Let's kind of make that jump now. Let's go into those organisms who got digits, but let's talk about that in between part. Let's talk about what went beyond the lungfish. Well, lobe-finned fish, because lungfish are also lobe-finned. Back in 2006, a fossil was discovered of the organism that looks, we believe, on the right-hand side called Tiktaalik. Tiktaalik was a lobe-finned fish that lived about 12 million years before true tetrapods, before true legged organisms. And you notice it's got lobed fins, and while it's not getting out of the water, you know, while it's remaining in the water, it's using those lobe fins for what we might consider an advantage, right? For example, if some of these lobe-finned fish, like the lungfish, develop lungs, well, the lungfish did, they could hold themselves up out of water. When water gets really warm, it holds less oxygen. So if the environment was warming up and the water was holding less oxygen, some fish can actually come to the surface and gulp atmospheric air. That's just gulp out of the air. Lungfish could come and do that as well, but if they could use those lobe fin to hold their heads above water, they would have an advantage. Tiktaalik looked like it was a pretty decent size, too. If there were prey running around on the shoreline, being able to come out like that would be an advantage, wouldn't it? Now, Tiktaalik died and stayed in the water, but Ichthyostega, there's Ichthyostega down below, which is an extinct amphibian, one of the very first ones, may have been one of the next steps, because it had these very sturdy forelimbs, but it moved kind of like a seal. Its hind limbs were more flipper shaped. So it would have been able to move around on land like a seal. Now, one of the other things that would have happened, and we see this in Ichthyostega, was when you move out of water, there is the desiccation problem, but there's also the weight problem, right? You weigh more, relatively speaking, out of water than when you're in water. It's the buoyancy part. So if you're Ichthyostega crawling around on land, you know, you're laying on your lungs and your heart, and that would restrict your ability to use those or even damage those organs. So what we see in Ichthyostega from these lobe-finned fish is further development of those ribs, making them broader, longer, stronger, allowing them to overlap to protect those vital organs when this organism moves out on land. Now, they were buccal breathers, these first amphibians, with lungs, okay? And buccal breathing basically is you gulp air and then swallow it. All right, that's not the way we breathe. We'll talk about the way we breathe. Well, that's in the other slides or the other recording that you'll listen to. But we're thoracic breathers. We breathe through our chest, okay? All right, so tetrapods are evolving. What other things are going on? There's Tiktaalik. Tiktaalik is one of these great transitional fossils because it's got so many characteristics in between. It's got scales, fins, gills, and lungs. It didn't mess around, so it may have been pulling itself up there to get a gulp of air. Now, among the tetrapods, necks, strong rib structure, the skeleton of the fins, right? The fact it was getting lobe finned, the flat skull and the eyes on the top of the head. You want to look up, if you will, as you're coming up out of the water. So see, this is like Archaeopteryx, isn't it? Amazing discovery, 2006, like 10 years ago. So we are finding those transitional fossil forms. Remember I said early on they used to say, where are those fossils that are in between? And we said, well, they're there. Tiktaalik maybe didn't live very long, right? Tiktaalik didn't leave behind an awful lot of offspring. So if you look in the fossil record, it took you to 2006 to find one of these, or a big enough one. All right, so there we are with Ichthyostega. And down below, again, is just a sort of a Ichthyostega coming up on a piece of floating debris there that might have been good, again, for breathing, right? Or getting out and attacking prey. And down again, I show you once again, look at the lobe-finned fish and look at the early amphibian, right? Lobe-finned fish, we start the structure. All amphibians did was kind of increase the strength, the size of the different, the shoulder, the humerus, and all the parts of the leg. And Tiktaalik, which I've inserted down below, would have been just about in between. So you can see there's a gradual change, maybe gradual on how fast that happened. It might have been more like cladogenesis. It might have been more like a very quick speciation process. But of course, once you got legs, you can support your body weight, especially out of land. You're going to develop lungs, right? Because they're much more, you can't use gills, even though they're more efficient. They don't work in air. They stick together. You're going to get a much bigger circulatory system. You're going to stick with a closed circulatory system. You're now going to add another loop to your circulatory system, which you'll see in the other recording. You're going to have a pulmonary loop that pumps the blood through your lungs, then back to your heart, then through your body. You're still tied to your water life, right? Amphibians, the word amphibian literally means a dual life. Amphibians live part of their life on land and part of their life in water, right? They got to go back to the water to lay eggs. And the reason why is their eggs, like you see in the lower right, the little things with the black dots, are anamniotic. Now, our eggs, bird eggs, have an amnion. Reptiles have an amnion. It's a sac around the fetus so that the fetus doesn't dry out. Inside of it, there's amniotic fluid. When a human female is getting ready to give birth, we say her water broke. Her amniotic sac ruptured, the amniotic fluid flowed out, and now it's time for Junior to be born. If you don't have an amniotic sac, your fetus dries out. Amphibians don't create an amniotic sac in their eggs. That's why they're called anamnions. So guess what? They have to lay their eggs in water. That's the reason why they have to lay their eggs in water, so they don't dry out. Stops them from desiccating. All right, now, since we're on this whole idea of legs, let me finish up and talk a little bit about legs in general. We see two creatures alive today, but one of the things we noticed in evolution is organisms that went from a more sprawling gait, like Ichthyostega, and kind of way like today, alligators still walk. We start to see organisms to a more erect gait. Well, why did this happen? Why did this happen? I'm Italian there for a second. Why did this sprawling gait evolve in some lines to an erect gait? Well, it comes out with something called Carrier's constraint. Carrier's constraint is a physiologist who figured this out. An organism with a sprawling gait, when it moves left to right, left to right. So what it's doing is when it's moving left, it's compressing that lung on the left side, which cannot fill as much. And when it's moving to the right, it's doing the opposite for the other side. So when sprawling gait organisms move, they're not getting quite the lung capacity it could get if they were erect. Because when you're erect, like you and I, we don't walk that way. So our lungs can completely fill every step. These guys are limited by what step they're taking. Again, if we look at this and we say, is one better than the other? One's better than the other for that particular organism in that environment. Because quite clearly there are still sprawling organisms around today. Like as I mentioned, our buddy the alligator. Okay, all right, now, this is where the part 3 of the recordings that are going to be posted on Blackboard is going to pick up. It's going to pick up with the amphibians. It's going to go through the amphibians. It's going to go through reptiles. It's going to go through the birds. Then it's going to stop. I'm going to do the mammals tomorrow in class, all right? So this recording right here, make sure I don't confuse myself. I'm looking at the way I have it. I've got part 1. This will be part 2. And then I'm going to say listen to part 3. Okay, so part 1, I'm going to post this as part 2. Then there'll be a part 3. Listen to part 3. And then tomorrow I'll finish up with this entire chapter with the mammals, okay? Does that make sense? And I'll label them part 1, part 2, part 3, okay? Okay, so let me stop the recording.